This episode is supported by two companies invested in advancing GPCR research, GeneTex and Eurofins DiscoverX. GPCR antibody reliability has been a bottleneck for years. GeneTex is working on changing that through rigorous validation and through collaborations with you, the research community. Request your favorite free anti-GPCR antibody sample and test it in your assays. Eurofins DiscoverX supports GPCR drug discovery programs with validated assay platforms trusted across discovery and development. Links to both are in the show notes. And now, let's dive into this episode. Hello everyone, this is Yamina from Dr. GPCR, and today I'm very excited to have with me not one, but two fantastic scientists who work at one of our strategic partners, the company called GeneTex. Today I have with me Dr. Chia Lin and Dr. Alex Ball. Welcome both of you to the podcast.
Hello everyone.
Thank you very much.
Good morning. Very good to have you on. So how about we start with you, Chia, and then we go to Alex. Can you please introduce yourself and tell us what it is that you do at GeneTex?
Okay, so hello everyone. I'm Chia. So I'm currently working in the GeneTex International Office in Taiwan, and I'm running the sales and marketing department, which involves sales, marketing, tech support, customer service, and also logistic team. So I foresee all of these activities in GeneTex and also doing activities for branding, doing activities for business development, and etc. So, for example, like this kind of activity coordinated with GeneTex, it's like initiated in the team. So that's me in the GeneTex. Thank you.
Alex.
First of all, Yamina, thank you very much for setting this up. I just wanted to say thanks to you and for Dr. GPCR for facilitating this podcast so we can introduce GeneTex to the GPCR researcher community. My name is Alex Ball. I'm a senior scientist at GeneTex. I've been with GeneTex since 2012. I really have a sort of similar to Chia, sort of a multifaceted responsibility set, if you like to say. I attend conferences, vendor shows, various meetings representing GeneTex. I review all the marketing information and literature in English that goes out from the company. And then also I'm involved in, exciting part, target selection, since GeneTex has antibody production capability, and we'll talk about that. And also the review that comes that our QA team deals with. So sort of several things.
And there's also the science in there and having those conversations with the scientists. For those who will be watching this podcast or are watching this podcast, when you send a request through Dr. GPCR to get a free antibody sample, and we'll talk about it, the email actually lands in Alex's inbox. So now you have a name and a face to knowing who will be working with you to get that sample in your hands for testing. So maybe, Alex, you can comment on what was GeneTex like when you arrived and how has it changed since? And then, Jay, we can go to you as well. I want to hear your perspective.
So just to give a little background, GeneTex was founded in 1997, and actually it was founded by eminent scientists in cancer biology and infectious disease, and it started in San Antonio, Texas, hence the GeneTex, out of a small building, and it was first a mechanism that sort of arose out of the desire to adequately distribute very well-cited and excellent antibodies that came out of the labs of these researchers. And then it sort of expanded into actually a business. The company then went on to, develop its own antibodies for a while. And that's primarily took the format of rabbit polyclonals, some hybridoma mouse monoclonals, and then also some licensed antibodies to build up the catalog. So I guess so that was kind of how it was. GeneTex was continuing to grow. The U.S. office moved in 2009 to Irvine, California, from San Antonio. Since I started in 2012, right about that time, you were starting to get the first inklings of papers that were coming out in major journals about the antibody validation, crisis and reproducibility and that kind of thing. And we'll talk about how with GPCRs, that was, really sort of highlighted. But GeneTex did a great deal of investment and shifted all of our new production, even though we had very successful polyclonals and hybridoma mouse monoclonals, shifted to recombinant monoclonal antibody production. And we tried to, dovetail that with a paper that came out that described a 5-pillar approach by Lin and Ball in 2016 to more systematically validate antibodies, which are very important for more challenging targets. So we've really seen, and GeneTex has continued to grow additional facilities, but more and more antibodies made in-house, and all of our new production since 2019 or so has been recombinant monoclonals with these improved and enhanced validation protocols.
Thank you. Jay, maybe have your perspective. I'm curious, how long have you been with GeneTex, and how did you see that evolution?
Yeah. Okay, so just to echo what Alex has said, so I entered GeneTex at the year of 2016. It's where the discussion of the science community regarding antibody validation and this reproducible crisis. So at that time, I'm at the marketing department, but we are, we need to prove ourselves to the science community that we are making antibodies, but with superior quality. So at that time, we realized actually making antibodies are, of course, need some technique, but to verify them and characterize them, or let's say validate them, let's actually take more effort, and that has to be based on science. So we read a lot of literature, we check a lot of information just to do our best to gather scientific information that could support our antibody, that we're using the most suitable materials to test this antibody. So that's what I when 10 years ago I entered GeneTex. So at that time, our company had a slogan saying quality antibodies, quality result. So that's how the researchers, I'm a scientist as well, so I'm a stem cell biologist when I do my PhD. So I knew how these not so optimum antibody that actually will waste researchers' time. So let's shift the whole company in 10 years. We're moving from polyclonals to monoclonal and recombinant, and quality is the thing that lies on the center of our products. So that's what I would like to share at the moment. Yeah.
I want to take a second to ask you a favor. A lot of you tune in every episode, but haven't hit subscribe. If you enjoy these conversations, please do it now. And if you have a moment, a review helps others find the podcast. And speaking of community, a couple of things worth mentioning before we continue. The Dr. GPCR University exists for those of you who want training that keeps pace with how the field actually evolves. Expert-led, live and on-demand, and built to serve discovery. And Terry's Corner with Dr. Terry Kanakin is where the frameworks that matter in drug discovery and GPCR research get examined seriously. With Dr. Kanakin in the room. Not content to consume, a place to think, to get. Thank you to GeneTex and Eurofins DiscoverX for being part of this community. Sign up for the Terry Kanakin Brief newsletter to stay in the know about upcoming live AMAs, live courses, and on-demand lessons. And join the Dr. GPCR ecosystem to stay in the know on upcoming live and on-demand masterclasses, community updates, and so much more. All the links are in the show notes. And now, back to the conversation. I love it. And I was going to ask, because I know I figured out that you're both scientists trained, and I wanted also to ask you what drew you towards antibody science. And Jay, I'm curious how a lot of people who listen to the podcast are junior scientists who might be at that cusp of what do I do with my life? And you're the proof that you can do a PhD and then in science and then move towards marketing in the life sciences. So I want to hear a little bit more about what drew you towards the antibody science. Let's start with you, Chia, and then Alex, if you want to chime in afterwards.
So, excuse me. So I did my PhD in University of Edinburgh, and I'm doing stem cell biologist and doing my postdoc over there. And at the end of my postdoc turn, I just start thinking of, what should I do? What's my career path and what's the plan at the moment? So there's a job that is quite popular 10 years ago, is scientific communication. And I like to talk. So I said, that's probably the good job that fits my career life. And at that time in Taiwan, this kind of job it's not that popular. But fortunately, my colleague, like my classmate at GeneTex, like at that time, he recruited me, say, "Oh, Jai, we had a marketing job that's also kind of scientific communication because I need to read literature and deliver that to either newsletter, posters, or like flyers that describe the science behind these products." Oh, perfect! That's what I could do, and I'm willing to do. And that starts my collaboration with Alice because Alice need to proofread all the material I prepared. And the first few times I'm pretty stressed because I just get the like total red of my writings. So but that's good. Like as a junior scientist at that moment, and also I'm not that familiar with industry. This kind of job is actually bridging from work in a lab to work in a company. So and that's what bridging me to antibody science. Actually, I'm doing some knockout mice when I was doing my PhD. So I spent 2 years doing the knockout. We didn't have CRISPR technology at the moment. It's about the time. So I do like traditional homologous recombinant. So I did a knockout mice, but there is no antibodies for this. So and I tried to purchase like one or two, and I didn't get any like confident result using my knockout mice. So it's like just say, oh, how can a vendor selling product that's didn't have good signal using the knockout license? So I'm a bit confused at that moment. So then when I entered this antibody industry, so we spent a lot of effort making our own knockout cells in-house, and we also collaborate with researchers just to so that echoes what we were saying at the beginning, the 5 pillars of antibody validation, like including knockout, knockdown. It's the center for antibody quality assessment. That's it. That's how I became from a scientist to a marketing and sales people in this industry. Yeah.
Thank you for sharing that. Alex, you're smiling.
No, I mean, Chia has such this wonderful trajectory for her training. Mine, not so much. Buckle up. So basically, I'm not something. This is not something that I would ideally recommend, but I went to medical school and I got a medical degree at the University of Southern California School of Medicine. Did a residency in internal medicine and really decided. I think before I started, my goal had always been to do research and at some point. But I decided. I just wanted to make that transition at that point. I wasn't sure that clinical medicine was for me. But I'm very grateful for that experience because I think it puts a different spin, a different take on science. I then went and worked for many years in a research lab, and the research lab was a lab that made its own antibodies, and you would learn the sort of vagaries, I guess, of what comes out after you inject a rabbit, and some antibodies, one antibody, same immunogen. One works great; it's the greatest antibody ever. The other maybe does a so-so Western blot. So it really sort of showed the how challenging antibodies could be because the science absolutely depended on it. Nothing would happen in the lab without reliable antibodies. And so I got a lot of bench research experience, and then it really impacted or really accentuated to me the importance of antibodies, not only for academic research, but even more so we've seen diagnostics and now therapeutics. I mean, the number of therapeutic antibodies that are in trials or up for is soaring. And so when this position became available at GeneTex, I was able to apply and luckily get it. And I have a great deal of respect for the founders of this company, and so it was really an honor to be selected. And I don't forget that on a daily basis.
Thank you, Alex. Can I ask? Actually, I don't think we named the founders originally of GeneTex. Can you tell us a little bit more about them?
Yeah. I refer to them as Dr. Lee and Dr. Lee, Dr. Wenhua Lee and Dr. Eva Lee, eminent researchers in cancer biology, cell cycle biology. They've been profuse. They were profusely productive both had high-powered labs. Dr. Joel Baseman, who was longtime head of the microbiology and immunology department at the University of Texas, San Antonio Health Sciences Center. He worked on Mycoplasma pneumoniae. And then Dr. Osborne, a clinical oncologist, helped was a founding member of the Baylor Comprehensive Cancer Center. His focus was on breast cancer. So we have a lot of people to look up to in the company.
I love that. Thank you for sharing that. First of all, I didn't know that the tax from GeneTex came because of the fact that the company was founded in Texas, which I think is really cool. Number one. Number two, what I find very fascinating is when basic scientists or scientists working in the lab identify a need and they decide to put together a company in not only to help themselves and the collaborators, but to expand and help the not the GPCR, but the community, the scientific community at large, which I think is hard, but at the same time very rewarding, because the impact that two scientists or a founding group of scientists can have by forming a company and building or helping to build the foundations of that company, you can't really measure it, but you know it's there as an impact.
Exactly.
And I think that's important.
Right.
All right. Going back a little bit to GeneTex, and considering that you've been at the company for quite some time now, are there any specific targets or projects that crystallize for you how difficult it is to develop GPCR antibodies? I see you both smiling, so Chia, let's start with you.
I would just leave this opportunity to Alice. Alice has an interesting story regarding this.
Yeah. So, I sort of, we talked about this, some of the upcoming podcasts, and mentioned this question. And my boss, Alan Lee, our president, sort of got pale, and he said, "Oh, LGR5." And I remember this was an issue that started not long after I started with the company. We wanted an antibody for LGR5. I don't have to describe this protein to this audience. I'll briefly state it's a key protein in adult stem cell maintenance, particularly a lot of focus on the crypts, intestinal crypts, in terms of generating the epithelial there. And it's been shown to be vitally important. LGR5 expression status is very important for a number of malignancies. And so it's of immense importance. So at the time, Alan, we got all the commercial antibodies we could get against LGR5 and tested them, and none of them were really met our expectations. And then, we struggled with our own, and now we have a polyclonal and a recombinant monoclonal that are, that we think are pretty good. But, it's something that we're always anxious about. We always want to get feedback about it, like, how did this work for you? This is an issue we'll touch on later in the podcast, but, sort of reaching out to the community and going, okay, let us know if this is, because these are notoriously difficult targets. And LGR5 is the one that really sort of introduced us to sort of the GPCR issue.
And you didn't start out with an easy one.
No.
I was 12 years ago, there was a paper that came out, and the title was Structure and Function of LGR5 and Enigmatic GPCR Marking Stem Cells. So that it's a difficult target. But I'm glad there's people who are interested in working on difficult targets, because that's how we advance creating tools for that. Was there any moment early on, a conversation at a conference or a result in the lab that made you realize how widespread and wasn't, LGR5 specific, the antibody quality problem was in the GPCR field?
We could share some experience. So my first conference, like international conference at GeneTex, is Neuroscience Society for Neuroscience meeting, SFN, in 2018. And at that time, I'm standing at a booth, and just customer come to the booth and just say, "Oh, I had a target which is mu opioid receptor. That's a GPCR." Like, the researcher shared some stories with me saying that he had a knockout mice, and he also purchased several commercial available antibodies, but none of these, well, a bunch of these give positive signal in his knuckle mice. So at that moment, like we didn't have products for mu opioid receptor at that time, but that's just kind of like punch at me, like, oh, so that's how like actual scientists they had struggled about, and they came to a commercial antibody company just say, oh, I had the materials, I'm willing to help you to verify any antibody you could generate it as long as they can work on their systems. So that's my first impression for how difficult these GPCR antibodies, how difficult these can like how to make them, and also there is a demand for a researcher. They're looking for a good quality of research reagent. Yeah, that's it.
I agree, and I think it's a matter of partnering, and it's really nice to have a company who is willing to collaborate and have those conversations in order to help solve a problem for scientists. And I don't think there is no company in the world who can validate their antibodies on every potential scenario and every potential cell line and knockout and you name it. And it can be beneficial to both partners collaborating. So thank you for sharing that little vignette. I really appreciate it. So when it comes to validated antibodies for GPCRs, what do most researchers commonly get wrong when they try these antibodies? What are some of the things that you're hearing from people coming to you saying, oh, it didn't work? I've had stories where, the data sheet looked great, you get it, and then you can 5 more, and then everything goes to trash because nothing works.
Yeah, so I think I wouldn't say people get things wrong. I think what I think the main message for me is that I think this is actually a healthy thing, and having been on both sides of this issue, both at the bench and now in the industry, and I'll be very honest with you, you really don't understand antibodies until you're in the industry. I mean, and what I mean is the whole the whole gestalt of the antibody world. You don't really, so it's sometimes, it's something that I think people at the bench haven't had that experience. You really get it once you're actually, in the industry to some extent. But I think the key is, and it makes me look back and think that I was pretty naive about antibodies in many ways. And part of that is I think there needs to be education earlier on in training about how to choose antibodies given their importance. But I think the key that I would come back to is I should have engaged more with the company. I think you find out, is this reagent really something that's going to help me answer my biological question? And you're going to look at the applications, you're going to look at the data. One of the great things about GeneTex is we show all the data we have. But there are things, sometimes you'll say, okay, and I get this question quite a bit, is that, all right, I don't see a Western blot. Does that mean they didn't try a Western blot, or they tried a Western blot and didn't work? So, I mean, those are two different things, I mean, those are two different, and it might shape, when you decide, hey, is this antibody something that I want to look at further, or if is this something I want to, expend money on, that kind of thing. So I would encourage people. I think the thing to do is to not be afraid to reach out to the company. We're certainly encouraging this with these GPCR antibodies, so that we don't have people wasting their time, which is the most expensive thing. Their time, their students' time, their postdocs' time, their scientists' time. Starting with a reagent that's not gonna do what they need it to do. So I think that would be the main. It's not something wrong. It's just something that, sort of the trajectory I've had, sort of the experience I've had now has made it very clear. Don't be afraid to reach out to the company. Say, hey, companies a lot of times won't reveal the exact immunogens. It's sort of proprietary, but at least at GeneTex, we're not going to let you wander around in our desire to just keep everything secret. We're going to make sure you understand, make sure what's been done on this antibody, what this antibody, we have evidence this antibody can or cannot do. So that's what I would do. I would encourage people to engage with the company. And I think a lot of wasted time and a lot of wasted money could be addressed if that was more common.
I agree. And then emailing you, Alex, directly and asking you these questions.
That's right. That's why I'm on the, you know who to yell at now. If the next time you see me at a meeting, say, hey, you didn't get back to me. But
yes.
We prioritize researcher questions. We try to address those immediately.
Wonderful. In practice, when you validate GPCR antibody at GeneTex, what does that look like in practice? I know we've talked a little bit about the 5 pillars of validation, but in your own words, can you please describe to us what does that look like? Alex or Jay?
Okay, yeah.
Whoever wants to go.
Well, I mean, Jai's the guru on this. I'll start out, and she can expand on it. So the way we look at it, and Chia leads a very effective team, and to, characterization of antibody. I do want to highlight something you said earlier, Yamina, that basically, antibody validation, so to speak, is almost an infinite process, right? I mean, it's a binder; it's going to react differently depending on what you do with it, But I think that I like to use the word that I heard a scientist, an eminent scientist use, and it's more like antibody characterization. So antibody really starts with choosing the right immunogen. And with GPCRs, that's even the most important. We've relied predominantly on peptide immunogens. The failure rate is high, but you can control what you're immunizing the rabbit with. So, and why is that important? So the example that I use frequently is, these are families. There's a lot of homology between different family members within the GPCR superfamily. So CXCR1 and CXCR2, 2 chemokine receptors, have extremely high, like 75% plus identity. So you have to choose the immunogen properly. If you don't choose, you're already messed up. You're doomed before you shoot a rabbit. So we choose our immunogens very carefully. As Chia was mentioned, we incorporate 5 pillar. Where possible, we try to do KOKD and GeneTex for maybe the top 40 or top 50 GPCRs in terms of most popular targets, quote unquote. We've invested in having KO lines made because we feel that this is one pillar, but it's a major pillar. Because if you can't meet that threshold, you're done. Start over. But then what we try to do, and again using variants of the 5 pillars, that we try to do several things. And I'll be very honest. If you go to our catalog, you'll see that this is an ongoing process. For some of the antibodies, we just have a couple of Western blots. This is what we have at this point. You go to other antibodies like our GPRC5A, I would argue it's the greatest thing on the planet because we have done everything for that. KO, multiple applications, data. We show all the data. We detected endogenous expression in MCF7 cells. It looks great. I'm like, I'd buy this antibody. But what we try to do is, we try to prioritize KOKD where possible. And again, ongoing process. We'll use something called comparable antibodies, and that's one of the pillars also. So we compare our antibody that we made, and these are all recombinant monoclonal antibodies. We compare our antibody with another well-known antibody from another source, side by side. Do we get the same band? Do we get a better band? Do we get not as good a band? Is it cleaner? But more importantly, are we, since this antibody has survived in the literature, it's a good antibody, and so we use that as sort of a comparison for our antibody. So we do things like cell fractionation. So, you do a whole cell extract and then you do a membrane extract and you hope the signal gets brighter. It's sort of, and then, we try to look at endogenous expression. And we've had, and, Chia and her team have been really good at identifying endogenous cell lines where you can see endogenous expression of some of these GPCRs, which often don't express that much. And, and then you can also check the localization. Because again, GPCRs move all around. It's well known in the cell. But if they're not on the plasma membrane, you need to really think what you're doing here. We do orthogonal testing. This is particularly for tissues. We'll stain on a tissue and compare it to like RNA levels for that tissue. So we use HPA or things like that. The Human Protein Atlas, which is a great resource for these types of omics to be able to do comparable testing. And then, as I mentioned, we do application-specific testing. And then what we've also been doing are GPCR arrays. This is from a nice company called CDI Labs. They've expressed a lot of the human GPCRs on the surface of herpes simplex virion, HSV1 virions. So it stays in a membrane, so it's happy. And then you can probe those with our antibodies, and we've gotten some nice results with that, with the CXCR2 and the DRD2. Some really nice, showing that the antibody recognizes the GPCR in one of its conformations. And finally, when worse comes to worse, we rely on overexpression. But then again, to keep with this validation sort of thought, This is why we were excited to get these antibodies out into the hands, of the GPCR researcher community. They have the best. They have the best reagents. They have the knowledge. They are the experts. We are not. And so this is why we're trying to get it out in a no-cost format, so they can look at the antibody and say it's good, it's bad, it's good for this, bad for this. So this is how we're trying to move it forward.
I love it. Chia, do you want to chime in? Then I will comment on this if you want to chime in.
Just echo what Alex just sharing. We had to know how exactly we would like to use these antibody for. So that's better to design the experiment and also design how we select the antibody and how we characterize the antibody. So that's the main point for these GPCR are different to study their function or their expression. Just need to be logistically clear of what we'll use these reagents for. So that's we're trying, but we need the feedback from the GPCR expert. So we already had these products available, and we now just want to let the expert to test them on the, I would say, most suitable materials. That's it.
And you both make a great point. It's so important to be able to put it in the scientists' hands in order to test in their own system. And sometimes, okay, let me track back. To your point, Alex, you guys have done an amazing job at characterizing so many of these antibodies. When you and I were talking a while back and you were showing me the data, I was like, okay, what am I going to, this is so much data for one or two or for a couple of antibodies that we were reviewing at the time that I was like, oh my God, I don't think I've ever seen so much characterization data for an antibody across so many different assay types. And that is something very important, because when you look for an antibody to purchase to test and you see that it's been used in so many applications, it gives you confidence as a scientist that it will work, number one. Number two, being able to have those conversations with the community is very important. And that's why when people are going to be listening or are listening to this podcast, we invite them to check out GeneTex's website on Dr. GPCR and reach out to you. The catalog is enormous. And so there is opportunities to test all of these, the antibodies of interest. And then number three, I think, although to your point, when some of these antibodies have, and I'm going to use air quotes, only used to test in Western blot, that doesn't mean it wouldn't work in a different application. And hence, this invitation to the community to test it in various applications. The more characterization this antibody gets, the more beneficial it is for the community. And I think that's very important. And I love how you didn't use the word validated, but instead characterized, because nothing is really fully 100% in every application validated. It's just characterized.
Exactly.
So I'd be curious to hear about what has been driving the shift towards recombinant monoclonals, and does it change? What does it change for the researchers working on GPCR targets specifically?
Mm-hmm. Oh,
I could
share
some.
You want to do that? Yep.
Yeah. So using of recombinant antibody is kind of common in the pharmaceutical field. So all the pharmaceutical antibodies, they are recombinant. But for research market, recombinant just emerged in the past, I would say 5 years. So I don't need to say too much about what's the advantage for recombinant, but first of all, it's recombinant expressed. So there is no lot-to-lot variation. And once it's been characterized or validated for a certain application or species, then they won't change. So especially we've been sharing a lot of how difficult to make a GPCR antibodies. And we just want to get all these characterization process worth it. So it's good to use a recombinant antibody just so everyone are there's no lot-to-lot variation, so everyone are comfortable for using it for all the follow-up experiment. So that's and in addition, since we know the sequence for the antibodies, we could do antibody maturation. So we could just change some residues, and then we further improve the performance of this antibody. So that's the key message I would like to share with all the audience, regarding the recombinant antibody.
I think of them as photocopied, because every time you get the same clone instead of, playing roulette and wondering what are you going to get with the next batch of polyclonals.
Right.
Alex, you wanted to say something?
No, I just I think Chia labeled it out or laid it out very, beautifully. They're defined by the primary sequence, so it's there. It's in a plasmid. It's not going away. So you have, and it's scalable. GeneTex is capable of making grams of this. It just depends on the size of the bioreactor. Consistent performance, and it's inexhaustible. It's like you were saying. I mean, it's not going to go away. So the antibody's there, you have it forever. It's not like, oh, I'm out of the serum for the rabbit from that rabbit. So
Yeah.
now, it's we're good. So yeah, I think, and that, to be very honest, is the way that the commercial antibody market is moving. Even though you still see a lot of good work with polyclonals, like Chai was saying, as you see more and more recombinants, and I think it's easier not only for industry, but I think more and more academic researchers who have to justify in grants and on their on their sub publications, they have to describe the reagents they're using. So recombinants are probably more favorably viewed.
No, I agree. I have a whole opinion around describing scientific data or reagents in publications, because oftentimes you look at it and sometimes you cannot find the catalog number in there, so that you go down this rabbit hole of figuring out, okay, what was the antibody or what was the reagent used. And having a specific catalog number and being very specific about it is also very helpful. And to our point, if it's recombinant, then if you buy it now or you buy it in 3 months, it's the same version, or the same copy basically of your antibody. All right, I have one last question, which is a little bit of a fun question. If you hadn't ended up in the antibody field, where do you think you'd be?
Yep. Oh, I'm first. Okay. Look,
everybody's looking at you.
I'm going to steal Jai's answer. I think scientific communication at this time is more important than ever. I think one of the things, interacting with so many scientists, having been at the bench myself, and having been, in medicine, you see that everything came off a laboratory bench. It really did. I mean, the cutting edge therapies. I mean, look what was done for hepatitis C virus. Look what was done for cancer. Look what's being done for autoimmune disease, metabolic disease. All of that was at a bench, at some point. Look at mRNA vaccines and these kinds of things. Everybody had to work out all this at the bench, and I think science needs to be a little more vocal about with speaking to society about, and it's been shown the economic value. It's like 2.5 times. Every dollar of at the NIH that's invested was like $2.5 come back in benefit. And so other than looking at the crass economic aspects of it, the reality is, I mean, you can't measure what it's done for for illness, for sickness, and for disease, and for prevention of disease, and improvement of health. I think scientists needs to really step up and slap themselves on the back and let people know that this has been this progress can't be taken for granted.
Agreed. Jay?
Yeah. So Ellen just steal my idea. So I think I could be a good scientific communicator to either write articles or just to transfer what we have done to the community. It's a very important job, to be honest. Like, scientists have done so much efforts to know how exactly the worlds are running, but then we need to let general people aware of what we have done, like to tell the truth and to spread the word. Like, we're not doing something that are irrelevant to the society. We're doing something like standing on the shoulder of each other's and to push the world to a better world. I would say that. So that's probably I would do it. Like Alice in different countries. Yeah.
Yeah, absolutely right. Yeah. And just last thing, just for GPCRs, a third of the drugs, like we're saying, are GPCRs. Of the FDA approved drugs are GPCRs. So it's untapped. It's an untapped. It's untapped science at this point.
Yeah.
Absolutely. I was doing some research for a prospective paper I'm writing. And so by 2031, not 3031, the market, the GPCR therapeutic market would be worth almost $7 billion.
Yep.
So lots of work to be done. But I want to also make another point here. Thank you both of you for sharing your perspective. You're right, Alex. Everything starts at the bench. And unfortunately, we don't communicate that enough. And I think it's because it takes a long time to go from the bench into the humans or into the hands of other scientists. And communicating it clearly and talking about these discoveries becomes crucial because it's hard to keep in mind. It's like when you're in the forest, you see the trees, but we need people who communicate, who are standing outside the forest and looking at the entire forest itself and tell people inside, hey, you're actually in a beautiful forest. Don't just look at the tree in front of you. There's many other types of trees around you.
Right. Great.
Wonderful. Thank you both. I really enjoyed getting to know you and getting to learn more about GeneTex. I appreciate you for your time and your effort and everything that you do for the GPCR community and the scientific community at large. We're going to now stop recording. Don't go anywhere. Thank you both.
Thank you very much.
Thank you.